Now these are the key exam questions on enzyme that you need to know before you go for your exam. I wanted you to get a pen, get a paper, and look at the question. Pause the video, read the question, and answer the question. Write the full answer. Then continue the video and watch my explanation and see and assess where you went wrong and how to get those extra marks that you have missed. So let's start with the first question. This is the graph and this is the question at the bottom. Describe and explain the differences between the two curves. This is a 5-mark question. So what I want you to do, I want you to have a go at the question. You can pause the video now, or I can read it for you and show you how to read a question. It's really up to you. So those of you who want me to read it for you, what's the technique of reading question? The first thing you do whenever you see a question, you don't try to read everything in one go. The reason because this will overburden you, cause you cognitive overload, cause you to be anxious, stressed, and you miss out important information. So what you do, just have an overview of the graph. So I can see a graph on the y-axis, concentration of product in a mixture, grams per decimeter cube. So this is my concentration of the product is increasing. Time after the start of reaction in seconds. So they have time here and I have how much I'm making in a reaction. So in other words, this is showing me how much I'm making, so the rate of reaction. I have two things. I have one of them is assessing at 25 Celsius and the other one is assessing at 37 Celsius. And obviously 37 showing a more rate of product formation compared to 25 Celsius. So now you have a overview, like brief idea of what the question is about, what the graph is showing. It's about temperature. Then have a read of the question. In here, when I'm reading, I'm going to underline, highlight key words. So a scientist investigated the effect of temperature, underline that, on the rate of enzyme control reaction. At each temperature, he started the reaction using the same volume of substrate. I'm going to underline that. And the same volume of enzyme solution, underline that. So in both of these reaction, 37-25, they both have the same volume of substrate and same volume of enzyme to making a control a fair test. Explain and describe. Describe and explain the differences between the two curves. 5-mark question. So pause the video and write your answer. Okay, so let's go through our answers. So this is the mark scheme. It's a 5-mark question. Initial rate of reaction is faster at 37. So if you look at this two curve here, we're comparing both of them. Now, it'll be incorrect for you to say that the rate of reaction is faster at 37 and slower at 25. That's incorrect because the first curve here, 37 Celsius, is, as you can see, it's not a straight line. So therefore, the rate of reaction does vary and it sort of slows down here. So it'll be incorrect to say. But at the beginning, as you can see here, this is kind of straight and it's much more steeper at the beginning compared to this for sure, for certainty. So you can say initial rate of reaction is faster at 37. Now you're explaining why this is faster, because there is more kinetic energy. That's why it's faster. Then you have to explain what does that mean. So there's more collision between the enzyme and the substrate, forming more enzyme substrate complexes. Then you are describing the second part of the graph where the 25 Celsius does not really plateau and 37 does plateau towards the end. So the graph reaches plateau at 37 Celsius. So it begins to go flat at 37, and the reason why is because all the substrate at this time, at this level, at this point is used up. We are increasing the substrate concentration, and when all the enzymes are occupied, when all the substrates are used up, then this is the maximum rate of reaction occurs. And I guess it can also be argued in this practical we have used same volume of substrate, same volume of enzyme. So you can also say at this level here, all the enzymes are occupied as well. Can give you the similar mark. That's why the graph plateaus around here. Let's move on to our second question. I have the same graph, same question, but in here I have two questions on calculating the rate of reaction. Number one, bit easy at 25 Celsius. The challenge is calculating the rate of reaction at 60 seconds for 35 Celsius. So I want you to calculate the rate of reaction of 25 Celsius. It's a straight line. And 37, I want a specific time. I want 60 seconds. So pause the video and have a go. Okay, let's go into our answers. The first one is asking you to find the rate of reaction at 25 Celsius. So whenever you look at finding the rate of reaction, the formula for any rate of reaction in a graph is y over x. So y, the difference in y over x. So in this example at 25 Celsius, because it's a straight line, you can use any point to find the rate of reaction. It's a straight line. So to make it easier, let's look at around here. So 240. So I need to find my y. So if I want to use this as my point to find the rate of reaction, then what I'm going to do is I'm going to basically draw a triangle if you want. So the triangle is quite difficult to see, but if you just imagine a triangle going down and a line going across all the way to 0. So I have my triangle here. Maybe if I try to draw this for you. Okay, so here we have our triangle. As you can see, our triangle for 25, and this is the formula, y over x. So you find y, so this is our y, so it's from 0 to 10, so our y is 10, and our x is all the way from there to there, 240. That gives us 0.04 gram per decimeter cube per second. That's our rate of reaction. Now, because it's a straight line, you can have the triangle done anywhere, and that will give you the exactly same answer. So try it out. So for example, I'll do it live here. I'm not sure if it's going to let me zoom out. So I have changed the triangle. Now I've done it here. So this is my y, which is 5. This is my x, 120. It should give me 5 over 120 should give me 0.04. So because it's a straight line, the triangle can be done anywhere. Now the second one is a challenge. Now the question said that you want to find the rate of reaction for 37 at 60 Celsius. Now it'll be incorrect for you to start drawing a triangle. The reason because the line is not straight, it's curved. So you have to draw a tangent. You can't use what I've done here straight away. Although at the end you'll draw a triangle, but you can't draw it straight away because the line is not straight. So a tangent. So this is how you do a tangent. So you draw a line that just is on top of, it has to be above your graph line, but it's just touching that 60, just touching it, and then you draw your triangle. So once you have drawn the triangle, you just follow the same equation. Rate equals y over x. So you find your y, you find your x, and then you divide. So my y is 9.5, my x is 90, my rate of reaction is 9.5 divided by 90 is 0.1 decimeter cube per second. So as you can see, the rate of reaction in a curved one, it always changes. So therefore you have to draw the tangent. let's go into our next question. Have a read of this question and then we'll go through the answer. Okay, so this is a very, very important question. Only 30% of students get full mark. It's a 3-mark question. Now, although you may not at this stage in year 12 AS level not done genetic mutation, you still should be able to access this. The genetic code determines the sequence of amino acids in a protein. Explain how a change in the genetic code can change the shape of a protein. So what you're doing now, you're going to highlight the keywords. So that the genetic code is talking about the DNA, determines the sequence of amino acid in a protein. So it's talking about the primary structure in the protein. That sequence of amino acid, which order it goes, is decided by your DNA genetic code. The question is asking, explain, meaning you have to give a scientific reason how a change in the DNA can alter the shape of protein. How a change in the genetic code can change the shape of the protein. And shape of protein, we are talking about the tertiary structure of a protein. Isn't here? Is not talking about an enzyme, it's talking about a general protein. If it was enzyme, it will specify it's an enzyme. I'm giving you an example here. This is a normal red blood cell here, and this is a sickled red blood cell here. And the reason why it's sickle is because the hemoglobin protein has changed shape. This is the help box here to help you to decide. So pause the video and have a go at the question. Write your full answer, 3-mark question. Okay, let's go through the mark scheme. Mark scheme, you have to mention that a change in the sequence of amino acid in the primary structure will be resolved. So if you change the genetic code, if you do a mutation, it will change the sequence of amino acid in the primary structure. So that's the first point. This change will alter the hydrogen ionic bonds in the proteins or in the tertiary structure. The reason I highlighted disulfide bond is because if you mention that, it should be ignored. Generally, in tertiary structure, we don't mention disulfide bond unless we are certain that there are sulfur within the amino acid that's being mentioned in the tertiary structure. So these two are enough. Because there's a change in the bonds, hydrogen and ionic bonds, this will alter the tertiary structure of the protein, and hence you have a different shape of protein. This is the 3 points that we should be mentioning here. In here, because it says protein, we just automatically think it's talking about enzymes, an active site, and we're talking about substrate. There is no mention about enzymes. Not all proteins are enzymes. So proteins is like a general term, and enzyme is a protein. But unless mentioned is an enzyme, you don't need to talk about active site, about substrate, enzyme substrate complex. Next question. So in here, this is a question. Have a read and then answer the question. I'm going to again tell you how to answer questions like this way. It looks like, whoa, this is a lot. So this is how to do it. So you look at the graph. Don't read the question. Just look at the graph. Y-axis, percentage of maximum protease activity. So protease is an enzyme. ASE is an enzyme that breaks protein. If you don't know that, look at the ASE. It's an enzyme. So as you can see, it's showing me the activity of the enzyme, how active it is. So the activity 100% here, 100. Okay, 80%. Okay, fine. And then it's showing me time in minutes. And then it's comparing the activity of this enzyme with 3 temperature, 30, 50, 60. So I'm thinking about temperature as a limit, as a factor affecting enzyme. And I'm thinking about kinetic energy here. After you have an understanding of the graph, your anxiety decreases. You think more rationally. You have more clarity. Then you start reading the question and underlining the keywords. So have a go at this question. This is the help box if you're stuck. Okay, so washing powders often contain enzymes from bacteria. These enzymes include proteases that hydrolyze proteins in clothing stains. The graph shows the effect of temperature on a protease that could be used in a washing powder. Explain, give reasons, the shape of the curve at 50 Celsius and 60 Celsius. So we can see here at 30 Celsius is sort of 100%, but as I increase the temperature to 50 and then to 60, the rate of reaction drops down. Now, what I want you to think of this is about your enzyme rate of reaction and temperature. If you remember, the line goes up, then goes down. The moment it goes down is because it denatures. So in here, although it's going, as you can see here, it's going down for both of them. So because it's going down, we know that the enzyme is being denatured because of high temperature. So explain the curve. You need to explain that 50 and 60. So let's see the answers. Both of them are denatured. As you can see, they're going down, they both are denatured. However, it occurs faster at 60. And the reason because, as you can see, at 60 is more steeper, so therefore is faster throughout, initially and towards the end as well, throughout. Generally is much more steeper than 50. So if you're very confident to say it's throughout, it's more faster. And then you're explaining, why does it denature at a high temperature? Because I said to explain. At high temperature, it breaks the bonds in the tertiary structure, and you can't just say bonds, you have to specify. So high temperature breaks hydrogen bond, ionic bond. Just don't say bonds, mention the bonds, and don't mention disulfide. Hydrogen, ionic, that's enough. This causes the active site to change shape, and if the active site has changed shape, that means it's no longer, keyword, complementary to the substrate. So therefore you have, very important, fewer enzyme substrate complexes are formed, so therefore the rate of reaction decreases or goes to zero eventually. I hope that was useful. Thank you for joining.